Good morning, everyone, and welcome. I'm pleased to have you here today. We're about to start a presentation on chemical sterilization. My name is Dr. S.K. Singh, and I hold a PhD in this field. Over the next few minutes, I'll be guiding you through the key aspects of chemical sterilization, including its principles, methods, and applications. So, let's get started and explore this important topic together. Let's begin by discussing chemical sterilization. This method involves using chemical agents, either in gas or liquid form, to effectively destroy microorganisms. It's especially important for sterilizing devices that are sensitive to heat and could be damaged by traditional methods. Some of the most common agents used in this process include ethylene oxide, or ETO, hydrogen peroxide, often referred to as VHP, and peracetic acid. These chemicals are highly effective and are widely employed across the industry. You'll find chemical sterilization to be particularly prevalent in the manufacturing of medical devices, where ensuring sterility without damaging delicate equipment is crucial. Understanding how these agents work helps us appreciate their vital role in maintaining safety and efficacy in healthcare settings. Let's take a closer look at the mechanism of action behind these sterilization methods. When these agents, like ethylene oxide, come into contact with microbial cells, they cause alkylation of DNA and proteins. This essentially means they add alkyl groups to these vital molecules, disrupting their normal functions. Additionally, oxidative damage occurs through agents like hydrogen peroxide and peracetic acid, which generate reactive oxygen species that damage cell components. These processes lead to critical outcomes: disruption of the cell membrane, inactivation of essential enzymes, and ultimately, microbial death. Understanding these mechanisms helps explain why these chemicals are effective sterilants. Now, moving on to the types of chemical sterilization, we see several options. Ethylene oxide, or ETO, is widely used because it can penetrate complex materials without damaging them. Vaporized hydrogen peroxide, or VH2O2, is another effective method, especially for sensitive equipment. Peracetic acid systems are also common, offering rapid and effective sterilization. And while chlorine dioxide has limited use, it's still part of the options available. It's important to note that roughly 50% of medical devices are sterilized using ETO, mainly because it's compatible with complex, sensitive materials that other methods might not handle well. This makes it a mainstay in many sterilization protocols. Let's take a closer look at the key process parameters that are critical for ensuring effective sterilization. First, temperature is a vital factor, especially for ethylene oxide, where we typically work within the range of 25 to 60 degrees Celsius. Maintaining the right temperature helps optimize the sterilization process without damaging sensitive materials. Next, gas concentration, measured in milligrams per liter, is crucial because it determines the potency of the sterilizing agent. Getting this right ensures that the microbial load is effectively eliminated. Relative humidity also plays a significant role, with an ideal range of 40 to 80%. Humidity levels can influence the sterilant's effectiveness and the overall process consistency. Then we have exposure time. This is the duration the items are exposed to the sterilant, and it must be carefully controlled to achieve the desired sterilization level. Similarly, aeration time, how long the items are ventilated after sterilization, is essential to remove residual gases and ensure safety. All of these parameters are critical because they directly impact the process lethality and validation. Proper control and monitoring of these factors help us guarantee that the sterilization process is both effective and reproducible. Let's take a closer look at the ISO 11135 requirements. First, it adopts a process lifecycle approach, which means we need to consider every stage from development through validation and routine control. Development is where we design the sterilization process, and validation involves specific phases like IQ, OQ, and PQ, installation qualification, operational qualification, and performance qualification, to ensure everything is functioning correctly. Routine control then ensures ongoing effectiveness. Key aspects of this process include bioburden assessment, which measures the level of microorganisms present before sterilization. Biological indicators are also crucial. They verify that the sterilization process is effective by using known quantities of microorganisms. Residual limits are set to ensure no harmful residues remain after sterilization, maintaining safety and compliance. All these elements work together to ensure consistent sterilization effectiveness. On the regulatory front, there's a global framework shaping these standards. ISO 11135 specifically addresses ethylene oxide sterilization, while ISO 14937 provides general validation requirements applicable to various sterilization methods. ISO 13485 focuses on the quality management system for medical devices, ensuring quality throughout the product lifecycle. We also need to be aware of the authorities overseeing compliance: the FDA in the United States, the EU MDR in Europe, and CDSCO in India. These agencies enforce the standards and regulations that guarantee safety and efficacy. In summary, these standards define the validation and control requirements that are critical to maintaining high-quality sterilization processes and ensuring patient safety worldwide. Now, let's take a look at the validation approach. When it comes to methods, we primarily rely on two: the overkill method and the bioburden-based method. The overkill method is quite conservative. It involves testing so thoroughly that we assume any contamination is effectively eliminated. The bioburden-based method, on the other hand, focuses on understanding and controlling the actual microbial load present initially. Moving on to qualification, we have three main stages: IQ, OQ, and PQ. IQ, or installation qualification, ensures that all equipment and systems are installed correctly. OQ, or operational qualification, verifies that the equipment operates as intended under normal conditions. And PQ, or performance qualification, confirms that the process consistently produces the desired results. Another critical concept here is the sterility assurance level, or SAL. Basically, SAL represents the probability of a non-sterile unit slipping through, so how confident we are that a product is sterile. The typical requirement is a SAL of 10 to the minus 6, which means there's only a 1 in a million chance of a non-sterile unit. This level of assurance is based on exponential microbial death kinetics, meaning we assume microbes die off at a predictable rate following sterilization. This scientific foundation helps us design and validate sterilization processes that are both effective and reliable. So, in summary, our validation approach combines rigorous methods, thorough qualification stages, and a scientifically grounded SAL target to ensure the highest level of sterility assurance for our products. Let's talk about hydrogen peroxide sterilization, which is a low-temperature process. This method is especially useful for sterilizing heat-sensitive materials. It's suitable for items like endoscopes and electronic devices, which can be damaged by higher temperatures. This process is governed by ISO standards, ensuring that the validation of VHP, or vaporized hydrogen peroxide, processes is consistent and reliable across different applications. When we look at the typical sterilization cycle, it often includes several key steps. First, there's preconditioning, where humidity and temperature are adjusted to prepare the items. Next is gas injection, where vaporized hydrogen peroxide is introduced. Then comes the exposure phase, during which the sterilant acts on the surfaces. After that, evacuation removes the sterilant, and finally, aeration helps eliminate any residual vapor, making the items safe for use. This sterilization method is used for a range of medical devices, including catheters, implants, and various plastic devices. Overall, hydrogen peroxide sterilization offers an effective, low-temperature alternative for sterilizing sensitive medical equipment, aligning with strict standards and ensuring patient safety. Let's walk through the calculation for SAL determination. We start with a bioburden of 10 to the power of 3, or 1,000 organisms. The required SAL, or sterility assurance level, is set at 10 to the negative 6, meaning we need to reduce the microbial load to 1 in a million. To figure out how much reduction is needed, we calculate the total log reduction. This is the sum of the reduction to reach a single organism, which is 3 logs, plus the additional reduction to meet the SAL, which is 6 logs. So, in total, we need a 9 log reduction. Now, if our d-value, which is the time it takes to kill 90% of the microbes, is 1 minute, then the total required exposure time is 9 minutes. That's because each d-value reduces the population by 1 log, so for 9 logs, we need 9 d-values, or 9 minutes. Let's clarify what the d-value actually is. It's the time needed to reduce the microbial population by 90%. For example, if we start with an initial population of 1 million, or 10 to the 6, then after 1 minute, which equals 1 d-value, the population drops to 10 to the 5, or 100,000. So, understanding the d-value helps us determine the necessary exposure time to achieve our target sterilization level, making it a critical parameter in sterilization process design. Let's talk about some important risk and safety considerations when working with ETO. First off, it's crucial to remember that ETO is both toxic and carcinogenic, so handling it safely is absolutely essential. We need to ensure that residual levels of ETO are kept within limits that comply with ISO 10993 standards, which are designed to protect both patients and staff. Additionally, environmental and worker safety regulations play a vital role here. These regulations are in place to minimize exposure and prevent health issues, so strict adherence is a must. It's also important to note that regulatory bodies enforce very strict exposure limits for ETO. This means that as professionals, we have to stay vigilant and ensure our processes are compliant with these standards to ensure safety for everyone involved. Overall, understanding these safety considerations helps us maintain a safe working environment while effectively using ETO in our processes. Let's take a look at the advantages and limitations of this sterilization method. Starting with the advantages, it's particularly suitable for heat-sensitive materials, which is a big plus when dealing with delicate instruments or plastics that could be damaged by heat. Additionally, it offers high penetration, meaning it can effectively reach all areas of complex or dense devices, ensuring thorough sterilization. Now, on to the limitations. One major concern is the toxic residues, specifically ethylene oxide or ETO, which can leave behind residues that need to be carefully managed to ensure safety. The process also has a long cycle time, which can slow down throughput in busy facilities. And finally, validation of this method can be costly and complex, requiring significant resources to ensure compliance with safety standards. Overall, while this method offers some clear benefits, especially for sensitive materials, it's important to weigh these against the limitations to determine if it's the right choice for your specific needs. So, to wrap up, chemical sterilization plays a crucial role in modern healthcare. It's essential for ensuring that our instruments and devices are safe for patients and staff alike. This process is also heavily governed by international standards like ISO, as well as national regulatory frameworks, which help us maintain consistency and safety across the board. Validation and ongoing monitoring are key components here. They make sure that sterilization is effective every time, and that patient safety remains the top priority. Looking ahead, there's exciting progress in developing safer sterilization alternatives, such as vaporized hydrogen peroxide and plasma sterilization methods. These innovations aim to make sterilization even safer and more efficient, aligning with the overall goal of improving healthcare safety and quality. For further support, please contact us. We're here to help with any questions or additional information you might need. Don't hesitate to reach out. We want to ensure you feel confident and well-informed. Thank you very much for your attention and participation today.